In 1951, the corridors of Johns Hopkins Hospital echoed with the footsteps of Henrietta Lacks, a young African-American woman whose visit would forever alter the trajectory of medical science. Born as Loretta Pleasant on August 1, 1920, in Roanoke, Virginia, Henrietta emerged from humble beginnings. Her life on a tobacco farm was cut short by the demands of supporting her family, leading to an early departure from school. In 1941, she wed David Day Lacks, and their love blossomed into a family of 5 children. However, at the tender age of 30, Henrietta faced a dire diagnosis at Johns Hopkins Hospital, an aggressive cervical cancer that would claim her life just months after doctors harvested her remarkable cells. Normal cells possess a self-destruct mechanism, limiting their division to about 50 cycles, a safeguard against the accumulation of genetic errors. This is known as the Hayflick limit. This process is a fundamental reason behind the natural aging of humans. Leonard Hayflick is a professor of anatomy at the UCSF School of Medicine and was professor of medical microbiology at Stanford University School of Medicine. His discoveries overturned a 60-year-old dogma that all cultured cells are immortal. Hayflick demonstrated that a normal human fetal cell population will divide between 40 and 60 times in cell culture before entering a senescence phase. This finding refuted the contention by Alexis Carrel, who is the winner of the Nobel Prize in Physiology or Medicine in 1912. Cancer cells, however, defy these constraints, dividing endlessly and overwhelming their normal counterparts. Yet, even these rogue cells typically perish outside the human body, reliant on the nurturing environment only the body can provide. Scientists cultivate human cells in laboratories to dissect their functions, unravel disease mechanisms, and evaluate treatments without risking patient health. Reproducibility and collaboration hinge on vast populations of identical cells capable of consistent replication over years. Before 1951, all attempts to sustain human cell lines faltered, until a world-famous cell biologist, George Otto Gay at Johns Hopkins, harvested the HeLa cell line from Henrietta's tumor. Mary Kubitschek, Dr. Gay's research assistant, extracted cells from Henrietta's tumor. These cells exhibited an uncanny resilience, thriving and multiplying endlessly in lab conditions. The team, recognizing their potential, dubbed them HeLa cells, a nod to Henrietta's namesake. Dr. Gay had stumbled upon humanity's first immortal cell line. Samples were dispatched globally, and soon the world's inaugural cell production facility was churning out trillions of HeLa cells weekly. These cells became the workhorses of medical research, albeit amidst ethical quandaries. The HeLa cell line revolutionized medical research. These cells were pivotal in countless breakthroughs. HeLa cells played a crucial role in the development of the Salk polio vaccine. Dr. Jonas Salk was an American virologist and medical researcher renowned for developing one of the first successful polio vaccines. His approach used a killed virus that could immunize without the risk of infecting the patient. This was contrary to the prevailing scientific opinion of the time, which favored the use of live viruses in vaccine. During the early 1950s, when polio was a significant health crisis, Dr. Jonas Salk developed a vaccine that needed to be tested for its effectiveness. HeLa cells, which are known for their ability to grow rapidly and indefinitely in a lab environment, became an invaluable tool for testing the vaccine. Dr. Salk's vaccine was tested in a massive trial in 1954, and the results, announced in 1955, confirmed that the vaccine was safe and effective. The widespread use of his vaccine led to a dramatic decrease in polio cases worldwide. Notably, Dr. Salk chose not to patent the vaccine or seek any profit from it, ensuring it could be distributed globally. In 1985, scientists utilized HeLa cells to discover that HPV, the most common sexually transmitted disease, can cause cervical cancer. Notably, Harald Zurhausen was instrumental in identifying the link between HPV and cervical cancer and in the development of the HPV vaccine. Harald Zurhausen was a German virologist who made groundbreaking contributions to the understanding of cancer. His research in the 1970s and 1980s led to the identification of HPV types 16 and 18, which are responsible for the majority of cervical cancer cases. For this work, Harald Zurhausen was awarded the Nobel Prize in Physiology or Medicine in 2008. HeLa cells have been part of several space missions to study the effects of zero gravity on human cells. In the early 1960s, HeLa cells were sent into space aboard the Discoverer 18th satellite. From these experiments, it was discovered that HeLa cells divided even faster in zero-gravity condition. This research was significant for understanding how human tissues and cells might behave in space, which is crucial for the planning of long-term human spaceflight and potential future colonization efforts. They've been instrumental in studies on AIDS, Parkinson's disease, leukemia, influenza, and the impact of radiation and toxins. Research involving HeLa cells has underpinned several Nobel Prize-winning discoveries, highlighting their impact on scientific progress. However, HeLa cells can also pose challenges and potentially ruin research results due to cross-contamination. They are known to be very hardy and can overgrow other cell cultures if proper laboratory practices are not followed. This has led to instances where cultures thought to be of different cell types were actually HeLa cells, which can invalidate research findings. In 1966, at a major conference of cell scientists, a geneticist named Stanley Gartler, who is Professor Emeritus of Medicine and Genome Sciences at the University of Washington, announced that the 18 most frequently used cell cultures, which were ostensibly distinct from one another, all exhibited a rare genetic marker that had only been found in African Americans. Since some of these cell cultures had been taken from Caucasian subjects, Gartler's conclusion was that all of the cell cultures had been contaminated with HeLa. Stanley Gartler's 1966 discovery revealed HeLa's propensity to contaminate other cultures, jeopardizing years of research. Their invasive nature, capable of hitching rides on dust or equipment, led to widespread culture takeovers. This contamination often went undetected for years, leading to invalid research findings and wasted resources. A notable scenario is the revelation that over 30,000 scientific publications in decades of medical research had to be re-evaluated because the cell lines used were contaminated with HeLa cells. Therefore, while HeLa cells are a powerful tool in research, meticulous care is required to prevent contamination and ensure accurate results. By 2018, HeLa cells had featured in over 110,000 scientific publications and nearly 11,000 patents, underscoring their monumental role in advancing biomedical knowledge and saving lives. Yet Henrietta's family remained uncompensated and in the dark for decades. Despite generating billions for biotech, Henrietta's family languished in poverty, unrecognized for her involuntary yet invaluable contribution. It wasn't until 1975, 2 decades posthumously, that the Lacks family learned of the HeLa cells through serendipitous research encounters. The struggle for recognition and compensation was arduous. In 2013, the NIH accorded the family control over Henrietta's genomic data. Rebecca Skloot's 2010 book, The Immortal Life of Henrietta Lacks, catapulted the family's plight into the global spotlight. The Lacks family had sued Thermo Fisher Scientific, accusing the company of profiting from HeLa cells that were taken from Henrietta Lacks without her consent. Thermo Fisher was one of the companies that continued to sell products derived from HeLa cells. The focus on Thermo Fisher Scientific in this case was likely due to specific actions and the commercial scale of their use of HeLa cell. In 2023, over 7 decades after Henrietta's passing, her descendants settled with Thermo Fisher, addressing the historical exploitation in medical research and underscoring the need for ethical integrity and patient rights. Cervical cancer, largely HPV-driven, is now preventable with vaccines and screenings. The silent onset of early cervical cancer underscores the importance of regular checks. With the 99% efficacy of HPV vaccines and routine Pap tests, cervical cancer stands on the brink of eradication as a public health concern. As we reflect on the extraordinary legacy of Henrietta Lacks, we are reminded of the boundless potential that resides within each of us. Henrietta's cells, which have traversed the globe and even ventured into space, continue to be a beacon of hope and innovation. They have not only unlocked the mysteries of countless diseases, but have also paved the way for a future where cervical cancer may no longer threaten lives. Today, as we stand on the cusp of eradicating cervical cancer, we owe a debt of gratitude to Henrietta Lacks. Her immortal cells have given us the tools to protect future generations from the very disease that claimed her life. It is a fitting tribute to a woman whose name will forever be etched in the annals of medical history. Henrietta Lacks, the mother of modern medicine, may her legacy inspire us to strive for a world where health and humanity walk hand in hand, where no one has to suffer from preventable diseases, and where every life is valued. Her cells changed medicine forever, and now it is our turn to change the world for the better. Henrietta's journey continues through each life saved, each disease conquered, and each step we take towards a more equitable and compassionate future.